Welcome to Lecture 8. This lecture will cover cell membranes as well as transport. Remember that membranes are considered to be a lipid bilayer. This means that they are mainly comprised of phospholipids, and these phospholipids are going to arrange themselves into two layers. Phospholipids are considered to be amphipathic. That means that they have a hydrophilic or polar head region, as well as a hydrophobic tail region. These phospholipids together will arrange themselves to form a bilayer, such that the tails or hydrophobic regions face in, and the heads or polar regions face out. Please refer to the diagram to the right to see this arrangement in the lipid bilayer. When we refer to the plasma membrane, we can say that it follows the fluid mosaic model. This is because the bilayer itself is very fluid and allows for components of the cell membrane to freely float throughout it. There are other components in the cells such as cholesterol, proteins, glycoproteins, glycolipids that will be able to move throughout the plasma membrane. We say that it is a fluid mosaic model because it is fluid as well as mosaic. When we say that it is fluid, we refer to the ability for substances or structures to move throughout the membrane. When we refer to it as being mosaic, this means that different parts of the membrane are going to have different components, such that one portion of the membrane is not going to be like any other portion. If we were to also take a look at the outer versus inner half of the membrane, they are also going to be different from each other. So if you refer to mosaic art, for instance, this will help to illustrate that concept. No matter which way that you look at the membrane, whether you are looking at the top of it, the bottom of it, a cross section, it is always going to be unique because of the way that these different components are distributed throughout the membrane. Let's discuss membrane fluidity. Lipids in the membrane are allowed to move within the membrane. However, this movement is going to be highly lateral. This means that flip-flopping between the top and bottom of the membrane layer of the membrane is going to be very difficult. In fact, it's not impossible. However, you need specialized enzymes that will allow those phospholipids to flip from one layer to the next. However, most of that movement is going to be lateral, meaning that within that bilayer, those phospholipids will be able to move amongst each other. Remember that there are double bonds that are present in one of the hydrocarbon tails. This will allow for a kink to be present, and this kink will allow for space between the phospholipids. This will help with the lateral movement. Remember that we also have cholesterol within the membrane as well. When cholesterol is inserted into the membrane, it will help to reduce fluidity in that area. However, when we get to very low temperatures, it will have an alternative effect, such that the high amount of cholesterol will actually help to increase fluidity at low temperatures. So cholesterol is very important in the cell membrane. This image here depicts a typical heterokaryon experiment. A heterokaryon is a man-made cell that exists from two different cell types. This type of experiment was very important in illustrating the concept of fluidity in the fluid mosaic model. In this type of experiment, a mouse and human cell were integrated together. The mouse cell proteins were stained in red prior to fusion. The human cell proteins were stained in purple. Once these two cells were fused together to form a hybrid cell, or the heterokaryon, the movement of these proteins were tracked over time. After approximately 1 hour, these proteins were randomly distributed throughout the new cell. This is important in illustrating that these proteins must have been able to move throughout the cell membrane. If they were restricted and not able to move, then you would see the overall distribution of proteins would be red on the top and purple on the bottom. However, because the red and purple are distributed throughout, this illustrates that all of these proteins could freely move throughout the membrane, thus supporting the fluid mosaic model. This image here depicts the different types of proteins that you can have in the membrane, as well as the cholesterol and distribution of the phospholipids. If you take a look at the lipid bilayer, you can see that there's many different proteins that exist throughout the membrane. You will also have cholesterol inserted in different locations of the membrane as well. You will have carbohydrates attached to both lipids as well as proteins to make your glycoproteins and your glycolipids. Keep in mind that you will also have the cytoskeleton features that lay right below the plasma membrane in the interior of the cell. So keep in mind that every section of this plasma membrane would be considered unique with respect to the different components of it, where the cholesterol is inserted, and also what is attached to it on both the intracellular and extracellular sides. There are many different types of proteins that you can have in the cell membrane. They can be classified as integral or transmembrane proteins. These proteins are going to be firmly embedded in the membrane. So these proteins are going to span through all the regions of the membrane, both the polar as well as hydrophobic or nonpolar regions. Peripheral membrane proteins, on the other hand, are going to be very closely associated to the membrane, but they are not going to be fully embedded. They may be either attached to a transmembrane protein, or perhaps they are only embedded in one side or the other, either extracellular or intracellular side. Proteins that exist on the inner part of the plasma membrane may also be anchored to the cytoskeleton. Proteins on the outer half can be anchored to the ECM or extracellular matrix. There are many different functions of membrane proteins. They can be used to transport various substances in and out of the cell. This is useful in your protein channels or protein pumps. They can also be involved in signal transduction, so allowing a cell to respond to or communicate with other cells in the environment. You can also have membrane proteins that can be enzymes. You can have various types of membrane proteins that help the cell to attach to the ECM or to attach to other types of cells, such as those membrane proteins involved in creating cell junctions. Proteins can also be important in cell-to-cell recognition. Keep in mind that there are many different carbohydrates in the membrane as well. However, these carbs are always going to be bound to either a protein or a lipid, forming a glycoprotein or glycolipid respectively. These carbohydrates are extremely important in marking the cell. These carbohydrate chains can vary and actually tell between species, individuals, or even cell types. An example of this is the different blood types that you can have with respect to your red blood cells. In humans, we can have A, B, O blood type. How we differentiate between these different blood types is by the presence of the different carbohydrates that these cell types have on their cell membrane. Another good example is an organ transplant. When a person undergoes an organ transplant, it is the carbohydrates on the transplant organ that can cause an immune response in the host. As a result, when a person undergoes an organ transplant, they typically first undergo cell type matching to ensure that you have a compatible match between the donor tissue and the host tissue. Another important component is to have the patient undergo immunosuppressant therapy. This is to suppress the host's immune system so that way the organ will not be rejected, meaning that the host's immune system won't attack the organ and deem it non-functional in the body. This is all simply due to the presence of the carbohydrate chains on the cell membrane itself. We will now take a look at the synthesis of membranes. Remember the endomembrane system with respect to the different organelles within the cell. The endomembrane system is also important in growing or shrinking the membrane. As vesicles are added to the plasma membrane, the membrane will grow. As vesicles are formed and as material is ingested into the cell, you will have the plasma membrane shrinking. So the balance of the endomembrane system will allow the cell to grow or to shrink. Remember that in the ER, this is where most of the lipids and proteins are going to be made. These are going to be transported to the Golgi, where any of these glycoproteins that are on the cell are going to be modified. Any lipids that are present in the plasma membrane are also going to be glycosylated in the Golgi. All of this material is going to be carried in a vesicle and then delivered to the plasma membrane. As this vesicle fuses to the plasma membrane, it will then become part of the plasma membrane itself. An important concept to understand is the concept of what is termed topology. This means that membranes within the cell have distinct faces or an orientation. If we consider the inside lumen of the ER, this would also be compatible or similar to the lumen of the Golgi. That means that if we have a protein that is floating around in the lumen of the ER, if that is packaged in a vesicle, it is also going to be in the lumen of that vesicle or inside compartment of that vesicle. That will then be part of the lumen of the Golgi as it travels through the endomembrane system, and then in the lumen of the vesicle itself. If this vesicle is then delivered to the plasma membrane, that means that whatever is in the lumen of that vesicle is then going to be secreted to the exterior of the cell. Similarly, if we have a protein that is embedded in that membrane that is traveling in that vesicle, that will then become part of the plasma membrane itself. If you take a look at this image here, this illustrates the concept of topology and protein trafficking. So we can see that in the middle of the lumen of the ER, we have these globular proteins. You can trace these proteins in the vesicle that then gets delivered to the Golgi apparatus. You can see that these globular proteins are still inside the lumen of the Golgi apparatus. When these are then packaged into a vesicle and delivered to the plasma membrane, these globular proteins are then going to be secreted. Now I'd ask you to refer to the transmembrane proteins. So this would be an example of how one of those proteins that is glycosylated gets into the plasma membrane. We can see that the transmembrane glycoprotein actually originates in the membrane of the ER. So anything that is transmembrane is also going to be embedded in the ER membrane. That is how it's going to be produced as a protein. That will then be also embedded in the vesicle membrane that delivers it to the Golgi. It would be part of the Golgi membrane. It would be glycosylated, meaning the sugar chains would be added to it. And note that the sugar chains are going to be added to the interior of the Golgi apparatus. So that means that the carbohydrate chain is going to be facing the inside of the Golgi, and subsequently it will also be facing the inside of the vesicle. Now pay attention to how this vesicle fuses. So as this vesicle with this transmembrane protein makes its way to the plasma membrane, we see that those proteins that were in the interior are once again going to be secreted. However, that transmembrane protein is now going to become embedded in the membrane itself. And take a look at that carbohydrate chain. That carbohydrate chain is then going to be facing the exterior of the cell. That means that whenever you have a transmembrane protein that is glycosylated, that sugar chain is always going to be facing the exterior of the cell. So all of those sugar chains are going to be on the surface of the cell membrane. Keep in mind that the cell membrane is also selective. That means that it is going to only allow certain substances to pass through it. In general, anything that is small and non-polar will be able to pass through the membrane unhindered. This is represented by substances such as hydrocarbons, lipids, steroid hormones, as well as small non-polar gases such as oxygen and carbon dioxide. That means that any of these substances will simply pass right through the membrane unhindered. Non-permeable compounds are compounds that are not able to readily cross the membrane, are things that are ions because they have positive or negative charges, or any substances that are polar. Remember, polar compounds will have slightly positive and slightly negative regions. So anything that has any type of charge will not be able to readily pass through the membrane. As a result, these compounds will need some transport proteins to help them across. Remember water. Water is considered to be a polar molecule. Why? Because oxygen has a high electronegativity value and it's going to be pulling the electrons closer to it, resulting in a slightly negative charge. As a result of this, the hydrogens are going to be slightly positive. By definition, this is a polar molecule. Because water is polar, it is not able to cross readily through the membrane. As a result, it is going to need specialized proteins that will help it cross the membrane. These proteins are called aquaporins. Transport proteins are going to be very important in allowing for these charged and polar substances to go through the membrane. There are two main kinds of transport proteins present in the cell membrane: channels and pumps. Channels are used for passive transport, so in instances where no energy is used to transport substances across the membrane, whereas pumps are used for active transport, so they are going to require energy in order to move substances across the membrane.